Hello and welcome to the Musella Foundation's Brain Tumor Awareness Month webinar series. Today's special guest speaker is Dr. Nick Vitanza. Dr. Vitanza is a pediatric neuro-oncologist at Seattle Children's. He directs the Vitanza Lab at Seattle Research Institute's Ben Town Center for Childhood Cancer Research, and he serves as Seattle Children's CNS CAR T cell lead and DIPG research lead. Dr. Vitanza, it's an honor to have you with us. Please take it away.
Thank you very much, Vanessa, and thank you for inviting me. Can you hear me okay? Okay,
Yep, we can hear you great.
great. So I'll be talking about intracranial CAR T cells for children with brain and spinal cord tumors, and I'll be honing in on a specific target, B7-H3. And I'm just putting this disclosure because B7-H3 CAR T cells were not something that I invented, but I ran the clinical trial, and it spun out into a biotech company that I'm now the advisor for. I'll talk about Seattle Children's pipeline and why we use intracranial CAR T cells here. I'll talk about this targeting of B7-H3 specifically, including a trial that recently completed and was published. And then I'll talk about some things that we're doing in our lab to try to make these CAR T cells more effective. So for folks who don't know, CAR T cells are a cellular immunotherapy that's targeted. And what that means is we're using a patient's own immune system to fight something. It doesn't even have to be cancer, actually, but in this case, it is. So what we're doing is we remove from a patient's blood some of their T cells, not an amount that they would miss, seen here. We genetically modify them to have a new receptor on their surface so that they can attack something like a homing signal. And you could really make them home into an enormous variety of things. We'll focus on a couple targets I'll talk about in a moment. And you have them target something on a tumor or another cell of interest. So now that T cell can bind. And what that binding does is it stimulates the T cell to eliminate that cell. And so you have a targeted immunotherapy against a cancer. Historically, this is most commonly done by, again, isolating T cells from the blood, engineering them. You grow them into the hundreds of millions and billions, and then you'd infuse them back into the blood. And that was initially because the first experience was with children with leukemia, who had very good responses to this therapy. And that was useful because that's where their tumor is, free floating in the blood for the most part. And also, it's just the easiest way to deliver these therapies is into the blood through an IV. Along with Children's Hospital of Philadelphia and Baylor had also developed these initial CD19 targeting CAR T cells for children with recurrent leukemia. And these trials started around 2012. I'm just showing the Seattle Children's trials here. But both Seattle Children's and CHOP published early data that there was dramatic responses for children with leukemia when they were getting CD19 CAR T cells. So children with leukemia that had failed other therapies, about 93% of the time were going into a remission, and a lot of those children, more than half, were becoming longtime survivors. Seattle Children's went through a strategy of modifying other leukemia targeting CAR T cells, seen here in orange. That was done mostly by Julie Park, Mike Jensen, and Rebecca Gardner. And then Naveen Pinto, Katie Albert, Julie Park, and Mike Jensen also worked on a solid tumor program where for children with solid tumors of the body, they were developing CAR T cells to these particular targets, still getting IVs delivered into the blood in this case, but to get to a solid tumor of the body. And when I came to Seattle Children's, one of the things I was tasked with was trying to translate this technology to children with CNS tumors using CAR T cells that for the most part were not ones I invented, although I'll talk about our own CAR T cells later, but bringing these technologies to children and primarily dosing them intracranially. At this point, there were no intracranial CAR T cell trials anywhere in the world, but Baylor had already started treating children with IV CAR T cells for brain tumors, and some adults had already done trials giving intracranial CAR T cells. So building off those combined experiences, we built this intracranial pediatric CAR T cell program. I won't go through a lot of the data of our first two versions of these trials, but they were called Brainchild 1 and Brainchild 2, and they were targeting two different things. Brainchild 1 was targeting something called HER2. It's most famous for being on breast cancer, but it's also on pediatric brain and spinal cord tumors like medulloblastoma and ependymoma. And then EGFR is on things like DIPG and other high-grade gliomas that we see. And so CAR T cells to these had already been developed. They had already been studied in the lab. We went to the FDA and got approval to run trials giving repeated intracranial dosing, and I'll talk about how we do that. We enrolled children who were 1 through 26 with any recurrent or refractory tumor except for DIPG, which I'll talk about separately. And they had to have a CNS catheter in place, and I'll talk about what that means. This is a very rough schematic of what a CNS catheter is, but Some people might be familiar with what a port is, which is an IV that goes into the systemic circulation and is more like of a permanent variety. An Ommaya is similar in that it's a plastic tubing that goes into one of the fluid bodies of the brain, and it comes up through the skull and takes a sharp angle, and then there's a plastic bubble that is above the skull but under the scalp. And so you could feel this bubble, which is under the skin, and you can put a needle into that, and because of the tubing is connected to fluid in the brain, so you could pull out spinal fluid or infuse things you wanted. This is not new technology. We didn't invent this. This has been around for decades. It just had fallen out of favor, at least in the United States, to be used for treatments. And now there's many more children with it because of the CAR T cell trials, I would say. The way that it worked to enroll in these trials is children would get screened, meaning we made sure that they really had a need to be on a phase 1 clinical trial. They came to Seattle and enrolled, and that was usually 3 days. The first day was talking about the trial. The second day was getting a central line placed to collect their T cells. The third day was collecting their T cells, removing the central line from the day before, and then they would fly home. Children with localized disease were enrolled on one arm where the catheter went directly into the tumor. For children with metastatic disease or lesions where you couldn't get the catheter into the lesion, they would enroll on an arm where the catheter was going into a fluid body in the middle of the brain, most commonly the lateral ventricle. So once we collected their T cells, we would then start manufacturing them and turning them into CAR T cells, and that takes about an average of 24 days, which is really 10 days to do and 14 days of quality testing. And then we built a paradigm where children would get repeated infusions. And I came up with this for a couple of reasons. One is that there had been some trials where children were getting one dose and that was the end of it. And we felt like that while that was working in animal models, which I'll show in a minute, it was just not representative of what we'd expect to see in patients. And because CAR T cells and T cells don't like being in the brain and probably won't last for very long, we would need repeated dosing. So while we could have run a study with one dose, make sure they're safe, that's the end of the study, we felt like we weren't really giving children a chance of success. And even though phase 1 trials are really geared at looking at safety, we wanted to have the potential for more benefit. The other thing is we built in that patients could dose escalate so that children who even on the lowest level would have an opportunity to get to more than one dose. So here's just an example of children starting in course 1, they get 3 doses at dose level 1, a week off, another dose at dose level 1, and then they would move up to dose level 2 for 2 doses. And at the end of those 2 months, that's when we would do the official evaluations to monitor for safety. So it was really 6 doses over 8 weeks. Initially, we had written the trials that children could go to 3 months, but as the trials were going and more children were getting to 3 months, we petitioned to the FDA and had the trial amended so that we could treat children indefinitely. And we published some of this initial preliminary experience in Nature Medicine and Neuro-Oncology. What we learned early on from these trials is that this was a tolerable and feasible approach is the short version. So this is one of our kiddos who enrolled when he was 2. He had an Ommaya that was placed, which you can see here. You can maybe tell the slightest bump, but can't really see when looking with him. This is him in the clinic, sitting in his mom's lap. We put some numbing cream on, let that sit for about half an hour. We clean the area. We put a butterfly needle into the area, which goes into this plastic bubble, so not into the brain. We can take off 10 cc's of CSF while the patient is just sitting there, again, outpatient awake. And then we can attach our thawed CAR T cells, give a dose directly through there. We flush the syringe and the tubing with normal saline. We pull out the needle, and the whole thing takes about 4 minutes. After that, children are observed in clinic for 3 hours, no mandated missions, and then children can go home after that. We didn't see more severe systemic toxicities, and that just means of the whole body, which is the main risk of when you're giving IV CAR T cells, is that patients have this huge immune response in their blood and can become very sick. It can even be fatal in some patients. CAR T cells, when given in the blood, at least when given into the brain, at least in our experience, they don't traffic to the blood. They stay in the brain. They degrade in the brain. They don't really get to the blood in significant amounts. And so we have not seen patients now with like 800 doses having any meaningful systemic, meaningful body toxicity. So the short version of our 2 initial trials is targeting HER2 and EGFR is that there had been no previous intracranial CAR T cell trials for children. We showed that we were able to make CAR T cells. Instead of one big dose in the blood, we could divide them into individual doses. We could dose children in clinic who were awake. This was all new information. So we were enthusiastic that this was a feasible process because this was really like, okay, now we have a ground floor that anything else we want to build upon, we can do. One of the diseases those trials didn't study and didn't allow patients to enroll with is diffuse intrinsic pontine glioma, or DIPG, which, as many of you probably know, is unfortunately an extremely aggressive, universally fatal tumor of the pons, which is an area in the center of the brain that you can't resect. Children get radiation, and of the about 400 kids per year, the median survival is about 11 months, and the median age children are affected is 7 years, meaning just this one tumor is responsible for about 25,000 years of potential lost life every single year just in the United States. So, along with all of our other devastating diseases, this is a universally fatal, devastating disease in and of itself. We didn't allow children on the first two trials, one, because we don't know if HER2 and EGFR are very meaningful targets for this disease, but more so because around that time in the mid 2010s, there had been a couple immunotherapy trials for children with DIPG that had closed early due to toxicity, and even some reports that children were passing away from immunotherapy with DIPG. So we wanted to get some experience with children who didn't have disease in such a high risk area, although certainly plenty of those kids still had high risk disease. But once we got some experience on Brainchild 1 and Brainchild 2, then we felt like we were better prepared to ethically and appropriately take care of these kiddos on immunotherapy. And so, starting with Sloan Kettering and Mark Sudane's team, and then St. Jude's team and Stanford's team, multiple groups had shown that this particular molecule, B7-H3, is highly expressed on a range of pediatric brain and spinal cord tumors. These are just 4 patients that I took care of with DIPG, whose tumors came to the lab and all looked like DIPG. This is just showing that 2 of them here, 1 and 2, have the histone mutation. Number 3 didn't have the histone mutation. Number 4 does. So these aren't all diffuse midline gliomas, which is having the histone mutation, but they all are what would be considered DIPGs, meaning tumors of the pons, and they all have very high expression of B7-H3. And that was important because then we took the B7-H3 CAR T cell that Mike Jensen had invented, and we started treating cells in the lab. We were able to show that with CAR T cell dosing, we were able to kill the tumor cells in a dish. When you're looking at even just the blue line is a very low dose of CAR T cells, and you can see you're stopping them from growing. If you're looking at these plots here, the gray line is if we give fake CAR T cells. So that's the rate DIPG tumor cells would grow at without stopping them at all in a dish. And so all of the other lines are just lower doses of CAR T cells. And then finally, at very high doses of CAR T cells, you can absolutely kill all of the cells. And we were able to show this not just against the 3 models that had the histone mutation, but also against PBT24, one of the models that came from a patient and was generously donated at the time of their biopsy, showing that the CAR T cells were affected even if you didn't have the histone mutation. And that's why on our DIPG trial, we don't mandate that children have a biopsy before they enroll, because from all of the evidence that we have, the cells should look equivalently whether you have the histone mutation or not. When we put the tumors into mice, which we do frequently, we're able to see that in one of our models at 100 days, where the mice are then succumbing to disease after that, when we give a single dose of CAR T cells into the brain, that's curative for the mice. And that is pretty consistent with a lot of preclinical, meaning laboratory CAR T cell experience, which is a double-edged sword in that it's very exciting. And in the beginning, people were presenting these laboratory experiments showing CAR T cells were eradicating disease, and everybody was very enthusiastic that a single dose would be curative for patients. And I think it was, as we looked at the adult experience, we really realized multiple dosing for children would need to be there to have a clinical benefit, and that our animal models, unfortunately, are not as predictive as we want them to be, which I'll also talk about in a little bit. But based on that, the clinical infrastructure we had built with Brainchild 1 and Brainchild 2, and based on the fact that targeting B7-H3 was very effective in the lab, we opened Brainchild 3, which had the same arm for children with localized disease, an arm B for the children with metastatic disease, and then a distinct arm C. And this was for children who have DIPG. And it was a similar protocol to enrolling, having your CAR T cells manufactured. Only instead of dosing every week, we would dose every other week. So instead of 6 doses over 8 weeks, these children were getting 4 doses over 8 weeks. And at the first dose level, we treated everyone at dose level 1 instead of going up. And then after we finished treating those patients, we went up to a dose level 2, a dose level 3, and ultimately a dose level 4. We're going from 10 million cells per each individual dose to 100 million, which I realize does not mean a lot. But this is trying to get to the maximum dose we can give, where we can safely give the CAR T cells in an amount that is meaningful against the tumor. And this is important because different than a drug that you swallow, you get absorbed, it moves throughout your body, it goes to most cells. When you're giving CAR T cells, this is a targeted immunotherapy that is having single cell interactions. And at a certain point, it is a numbers game. If you don't have enough T cells and you have many more tumor cells, then it's going to be a struggle. And actually, this is one of the problems with a lot of lab work that's doing CAR T cell work is that you implant a tumor and then you immediately give CAR T cells that way outnumber the tumor. And it's just not necessarily realistic of what children are going through. So that's why sometimes the lab results look artificially beneficial. But what the lab results are really showing is that these are biologically active T cells, and that even when they're outnumbering the tumor, they're not causing severe toxicity as they're acting. So, Brainchild 3, ARM C, giving B7-H3 CAR T cells to children with DIPG is what I'll hone in on now. One, because it is the group of patients that I spend the most time taking care of, and that what my lab focuses on, but also because children with DIPG have an unfortunately very uniform clinical experience compared to other diseases that medulloblastoma, ependymoma, ATRT can be much more variable with very long-term survivors, children who have tumors that grow very fast. At least for most children with DIPG, unfortunately, it's a very similar experience, which can help us better understand the toxicity and the benefit when we look at taking care of them. So again, Brainchild 3, we enroll children 1 through 26, trying to go as low as possible. Again, I have a frustration with when trials only go down to like age 3, 4, or 5. I don't really understand that. If you'd think it would work just as well for a younger age, the maximum we can treat at our hospital being a freestanding children's hospital is 26. Um, Performance scores are just a way we measure how well children are doing. For ethical reasons, we can only go so low. So we enroll children down to 60, which is generally when they could still participate in some quiet play, but are not participating in active play. And then the other thing that was important to me is that not to overly curate who we could enroll. So we allowed children with metastatic disease, not just with first radiation, but even a second radiation, and in one case, a third radiation before they come to us to try to reach as many people as possible because there's already so many inherent limitations to who we can enroll based on families' finances, travel, how many enrollment spots you have. So while the trial was open, we wanted to enroll as wide a group as possible. I will not get into the details of all of this, but I will say that this is what's called a consort diagram, where you talk about the children who are enrolled at each dose regimen, which is what DR stands for. So DR1 was treating at 10 million cells, moving up to 25 million cells, 50 million cells, and ultimately 100 million cells. I bring this up to point out, sadly, one patient we took care of who was 4 years old had bleeding inside her tumor after her first dose. That was our only dose-limiting toxicity. This can be confusing, but dose-limiting toxicities is actually not the same definition for each trial. So on some trial, you could get really sick and it doesn't get counted as dose-limiting toxicity. On someone else's trial, you might not get sick at all, but it's a very conservative place, so it's called a dose-limiting toxicity. On our studies, dose-limiting toxicity basically meant that you had a severe toxicity that lasted for more than 72 hours. And I won't get into more of the grading details, but can answer questions if people are interested. But this little girl had bleeding and unfortunately had to come off the study. But thankfully, we didn't see that in further patients, and we were able to get up to the highest planned dose of 100 million cells. As far as what's the toxicity we see. So children, again, this is outpatient. They're having their Ommaya accessed, which means the needles put in, some CSF taken, CAR T cells put in, and then usually somewhere in the course of, on the earlier side, 3 hours, and on the later side, maybe like 15 hours, children usually have headache, sometimes nausea and vomiting. Not uncommon to have transient worsening of a baseline neurologic issue. So if your arm is weak, it's a little bit weaker. If you're having trouble speaking, that can be a little bit worse, but usually resolves on its own without intervention. And for some of our patients who have had a lot of doses, like we actually have one little kid that I'll talk about who's had over 90 doses, and the more doses he's had, the less side effects he's had from them. This is actually MRI imaging of their tumor. So I met him when he was 2 with histone mutant DIPG. He's now 6. He enrolled on the trial after getting upfront radiation, has had now over 90 doses of every other week intracranial CAR T cells. And over the course of his period, he's had decrease in tumor size, decrease in T2 hyperintensity, and clinically now is what I would consider a physically normal 6-year-old boy who's participating in soccer and other activities. Unfortunately, that has not been the situation for every child we've taken care of. And so, even for children where we have seen a response with CAR T cells, it is not always sustained. So, there's definitely, unfortunately, some children who didn't respond. And even for children like this who did respond, this patient ultimately progressed metastatically with disease in multiple areas. So, we know that we have a long way to go to improve these therapies and really get children the treatments that they deserve. I won't go into each individual patient that we took care of, but I find it important to show as much as possible about every single kid we're taking care of when they're enrolled on these trials. This is all published and is free to be shared. But orange is essentially life before children got CAR T cells, blue is afterwards. So I think what one of the positives that we've taken away is that even with repeated dosing of CAR T cells, so for example, this patient here is the patient who's had 90 doses, and this data is actually 6 months old. All three of these patients with DAPG are still alive more than 4 years from their diagnosis. I won't get into survival too much because these are phase one trials. And honestly, while I know that that is the thing that even as a parent myself, I would ask about the most, but the numbers are so small, it's really hard to interpret. But again, we've been, I think, enthusiastic that it's been tolerable and unfortunately not reaching every trial that we want to, but there are some children who seem to be improving with treatment. This is just to show two things. One is that we want to learn as much as we can from every child who's giving so much and being so brave to enroll in these trials. And so, without putting them through extra procedures, whenever we do collect blood or do collect CSF, we just try to learn as much as possible from it. And one of the things that we've been focused on is what are different markers that go up in the spinal fluid after a child gets CAR T cells? And two of the main things that we've published on multiple times now is interferon gamma and IP10 or CXCL10. And that'll come into play in a second. And we see those spike in the brain after each dose. And so knowing that that's there and those things are being released, they're basically just molecules that can come out of T cells, come out of other immune cells in the brain, and I'll also show can come out of the tumor cells themselves, is how can we leverage that as we're building better CAR T cells and better treatments in the future? So from there, I think I've talked about what Seattle Children's overall pipeline is, the kind of ecosystem of focusing on targeted immunotherapy, the B7-H3 experience specifically, where we were able to finish a phase one trial and show what I think was progress for the field and benefit for some patients, but still left a lot of room to improve. And so in my lab here at Seattle Children's Research Institute, I'm very fortunate to work with an amazing team of people that's funded by an amazing group of philanthropic donors, the NIH and other groups, to try to improve these therapies and others against some of these tumors. And Dr. Edward Song, who trained at Penn and is a scientist in our lab, when he came to the lab from Pennsylvania, we were talking about what are ways that we can make better CAR T cells. And we had all this data of what chemokines and cytokines, these molecules that are secreted in the brain after CAR T cells, what they are. And so we looked at one of them that was going up the highest. We found the receptor that was associated with that, meaning like that molecule is released. What's the receptor on a cell that actually binds with it? And then we thought, okay, well, if we have a T cell, can we make more of that receptor on its surface so it binds to that molecule even more? And can this be a way that we can get a T cell to migrate better to the tumor? And so this is the overall hypothesis that we had is a group of T cells in green start getting near a tumor after a dose. They start to engage, they're secreting some of these chemokines and cytokines. The yellow receptor here is the T cells engaging with that, which brings them to that gradient and drives them that direction. Even more is secreted, and then you get this positive feedback loop of more T cells going to the tumor, which is a known problem in solid tumors that maybe you're dosing a lot of T cells that just don't get to where they need to be. So in the lab, we kind of modified this already existing technology called a transwell chemotaxis assay, where we could put our T cells into a little insert inside a larger well, and basically we could test how many CAR T cells migrate through there. And when we built CAR T cells that were overexpressing this receptor, which is here in red, more of them were migrating through this well. Okay, so that's great. They're moving better. That's encouraging. But obviously we need to kill tumor. So we want to make sure that during this migration pathway and that stimulation, they don't kill less tumor. And what we saw is this is an overall number of tumor cells. If you give our regular B7-H3 CAR T cells at a very low dose, you're killing about 20% of the cells in this assay. When you give the same dose of these CAR T cells that have this receptor on them, we're able to kill more like 50%. So a sizable difference in how much of the tumor we're able to kill when the tumor is at the bottom of this well. So not only are the CAR T cells migrating and with directional intent, but when they get to the tumor, they're able to kill it. And the other interesting thing we saw along the way is that this molecule CXCL10, which we know is secreted by T cells and other things, is also secreted by DAPG cells themselves. So this is these lines that look like they're all the same, but if you look at the numbers here on this y-axis, the numbers on the side, they start off in this range of 200, 400, 600. After 4 days, that's 2,000, 4,000. By the time you get to 1 week, we're in the tens of thousands. And that's the amount of that molecule that's getting made, not by the T cells, but actually by the tumor. So that if we can harness this, we potentially have a way to migrate directly to the tumor that's secreting things telling us where we need to go. To make sure that I leave time for questions, I'll just talk about this animal study and one other project that we're working on. But when we do this in the lab, it's not a perfect system for how we can get more information for how to perfect this therapy to get to a child. Unfortunately, a lot of things look really good in a dish and then have no benefit even when you get to a mouse or another animal model. And so we wouldn't want to run a $20 million phase 1 clinical trial that is money and resources that could be put to something else for children, and more importantly, treat children on a therapy that is not as good as it could be. So we do go through animal studies where we implant tumors directly in the brain, and then we give the CAR T cells either in the blood or the brain to see how they do. And so these are mouse images here. IV are mice that we're treating through an IV. So they have a tumor that's implanted, and then NTD means fake CAR T cells. When we give them fake CAR T cells, all of the mice pass away within 14 days. When we give them IV standard CAR T cells, they still pass away in 14 days. This is one of the reasons why we prefer intracranial CAR T cell dosing. But when we give our CAR T cells that have this receptor that helps them migrate better, more of the mice are alive at day 14. No comparison, though, to when we give directly into the brain, where if they're fake CAR T cells, again, the mice die at 14 days. If we use our standard CAR T cells, we're able to improve survival for most of the mice, but you can see like even a spinal cord lesion that's developed in this one mouse. But when we give our overexpressed CAR T cells, we see very little tumor at day 14. And this is just another way showing our mock brain CAR T cells, our standard B7-H3, and then our other ones for how much basically tumor size is left after we give them. And then before I was showing you cut off at day 14, but if you follow these mice for the rest of their life, in the group where they're getting our standard B7-H3 CAR T cells at a pretty low dose, the mice are all passing away around day 70. When we give our ones with the overexpressed receptor, more of these mice are alive. And we're really enthusiastic about this and hoping that overexpressing this receptor could be beneficial for CAR T cells going forward and helping them migrate to tumors better. I will call out, we are not the only group who's doing similar things. And Mike Verneris at Colorado, groups in Florida have done similar things with other molecules. But this is, I think, the first approach for DIPG and using this particular receptor. The other thing is, what's the potential for combinatorial benefit? I mean, taking care of kids in clinic all the time, I understand the enthusiasm for getting the combination therapies. Obviously, that's been the game changer for diseases like neuroblastoma and leukemia, is not just one home run agent, but multiple drugs layered together. And so we screened a bunch of drugs, and this is done by Dr. Andrea Timpanaro, another scientist in our lab, who looked at this agent ONC206. We screened a bunch of chemotherapies and another drug called ONC201. And unfortunately, they either weren't very effective in our models or they also kill CAR T cells. So you think general chemotherapy kills living things. That's why children get so many side effects. CAR T cells are living things. So a lot of chemotherapy also uses CAR T cells, which is why we can't just run a trial where we give regular chemotherapy and CAR T cells at the same time. The chemotherapy would kill the CAR T cells. So modeling this in the lab, we were looking for drugs that at least had some benefit, but didn't negatively affect tumor cells. And one of the things that came up with this drug, ONC206. And this is just showing the number of tumor cells that are alive, if you think of it as 100% here at this one. And as we go, each one of these circles is a slightly higher dose of ONC206 in different tumor models. We're able to show that at a pretty reasonable dose of ONC206, we're able to kill the tumor cells. So this falls into a category of drugs we think is worthy of further investigation. Without getting into all the details of this, this is showing that when we give ONC206 to CAR T cells, we don't kill CAR T cells or change the CAR T cells for the most part. And so these are things you could dose together. And then I'll take a second here and then also a brief movie and then show an animal experiment. But this is, again, the type of study where the data you're looking at is the higher something is going, the more cells are alive, and mock means fake CAR T cells. So when we put different DIPG cells into a dish, these are different models that have been generously donated from patient biopsies. You can see that when you give fake CAR T cells, all of the tumor cells grow over 150 hours. That's the red line. The orange line is if we give kind of a dumbed down CAR T cell dose. So we could kill all the cells if we wanted to, giving a slightly higher T cell dose. But giving a lower dose, we try to kill about half of them. And that's what you can see in the orange line. And then when we give the median dose of ONC206 that we think is tolerable, we're able to kill about half of the DIPG cells. So by itself, not sure how beneficial that would be. But when we put it in combination with our CAR T cells, which is the green line, you can see now these tumor cells are really tanking in their viability, meaning how many tumor cells are alive. This is just a video of that using the same color scheme. So this is fake CAR T cells. These are tumor cells. Yellow is when we give CAR T cells. Blue is just the drug ONC206. And then down here in green is combination. And I totally appreciate that you guys are like, Nick, we don't look at this all the time. What are we looking at? In the bottom right-hand corner, it just becomes a bunch of dots and debris, and that is cells dying. And then you can just believe me that that's true. And then when we do animal modeling again, this is showing the survival of mice that are dosed. When we look at the combination of regimen, mice were able to live longer. And now we're doing this in more and more models to make sure that this is true against different types of tumor, to look to see, are there combinations that we can bring forward in future clinical trials. I don't know that this combination itself is the answer, but it's a proof of principle, and I hope will be a roadmap for the field on how to test a targeted immunotherapy with other agents, and then how to properly assess what's the effect on a T cell, how does the T cell affect the tumor, what's the right dose to give these together to have the maximum benefit, and then be as safe as possible for a future patient. So hopefully that was an okay amount of time to leave questions. In summary, I'll just say what I think that we've been through a really robust infrastructure at Seattle Children's, a really supportive environment here. On the clinical side, writing these trials, enrolling the first group of dozens of patients, we really worked very hard to build what I think is a safe and reproducible system that now some other hospitals are using as well. There's also excellent programs at Baylor, Stanford, City of Hope, St. Jude, with others coming online and internationally, also in Rome and Sydney and London. So getting to work with these programs and build the future together has been really rewarding, and we're really thankful for how brave our patients have been. And across these trials now, just at Seattle Children's, we've given over 800 intracranial CAR T cell doses to about 150 kids who have enrolled. Our B7-H3 CAR T cells were safe in this completed trial for children with B7-H3 and are now moving into a phase 2 clinical trial. I'll mention that the FDA actually granted us something called breakthrough designation and something else called RMAT. And these are basically two things that the FDA gives when you finished a phase 1, they go through a rigorous analysis of your data, and they want to help you build a phase 2. And the other thing, which I think is super important, is one of the things I'm the proudest of, is our phase 2 trial is not going to have a placebo control. We're working with the FDA that we're comparing the results to historical controls so that we don't have to put children through a placebo medication. And that in the lab, CXCR3 overexpression seems to have some benefit, which again is this helpful migration. Combinatorial regimens, I think, is a long road, but we're excited about. And then I think the other thing that's a very important take home is this is very much the ground floor of what these things can do. This is different than like if you give a drug like an HDAC inhibitor or a ClpP agonist or something else, and you're like, well, that worked or it didn't work, and now there's kind of only so much modifying you can do this agent. These CAR T cells, we can engineer dozens of changes to them. We can give them in all types of different ways. We can play with the medicines we give first. And I think that for better or worse, we could spend the next 10, 20 years trying to optimize this for children, but this is very much the ground floor of what's possible. And we have this phase 2 trial, which we just invited several sites to and are hopefully opening in early 2026, if not late 2025. We're very proud to be working with an iterative, meaning we try to learn from patients and bring it to the lab the same way we try to learn from the lab and bring it to patients, and working with a bunch of excellent centers. And our team at Seattle Children's is amazing. Julie Park and Mike Jensen have been the best mentors you could ever ask for and gave me a lot of latitude, even as a young person, to bring my ideas forward. Not that I'm young anymore, but I used to be. And then Sarah Leary, who runs our neuro-oncology team and is amazing. Rebecca Ronsley, who's now the PI of our trials and does a wonderful job, and Allison Thompson, who's our coordinator. And then so many amazing organizations who have donated to us or reviewed our grants and approved us for funding, and also recently getting awarded NIH support for a clinical trial, which will hopefully be 7 years of funding to advance therapies for kids with brain and spinal cord tumors. And I think I'll leave it there. But thank you, everybody, for joining and for your attention.
Thank you so much. This was absolutely incredible, and we do have a lot of questions, so I'll get started with those. You mentioned at the very beginning that Omiya reservoirs had fallen out of favor for a while. Can you explain why? Were the risks misunderstood?
That's a good question. It's kind of complicated, and I'm not an OMI expert. It is a neurosurgery to place it. I would say neurosurgeons don't love when I say this, but it's on the bread and butter end of a neurosurgical procedure, usually in the hospital for one night. Insurance pays for these, so you don't need the clinical trial to. You tell, your insurance company, we need this in place to deliver a therapy, and insurance covers it. So that's been successful. There are other countries, especially in Europe, where even young children get these placed preemptively for things like medioblastoma or high-grade gliomas, and they do get intracranial, old-fashioned, maybe not the right word, conventional chemotherapies through there. We still do this in children sometimes, but in the US, we're more often to do it by spinal tap. We do know that if you give a spinal tap of a chemotherapy agent, it can move up the spine, but it doesn't coat the brain at a very high percentage. And the other way around, when you dose from the top, you don't get full coverage at the base of the spine. So I think that probably there weren't newer medicines to give through it, so it fell out of favor. But now, as we have new technologies that potentially have a benefit when you give directly into the brain, it's been convenient to kind of revisit that. They're still used for a lot of other conditions, but hadn't been used as much for pediatric brain tumors. Like I trained at NYU and then Stanford before coming here, and over like 8 or 9 years of oncology training, I had never seen a child with an Omiya before we opened our trial. And now, I know hundreds of kids.
Okay. On your slide about the brainchild 01 and 02, you said no CRS and no ICANS. What do those letters stand for?
Yeah, sorry, I got lazy and I just converted some academic slides to this and didn't explain everything. CRS is cytokine release syndrome. It's unfortunately a low incidence but fatal complication of getting CAR T cells for leukemia. Basically, you get CAR T cells in the blood and you get such a high amount of inflammation in your body that it moves to your brain and can be lethal. Can also cause other problems in your body. There was a worry when we first ran our trials. I will tell you, I went to conferences where actually other neuro-oncologists were saying this would be a very dangerous, fatal approach. But what we felt was that actually intracranially avoided a lot of the systemic problems. And I think while it's not perfect, we've seen that to some extent. ICANS is basically another syndrome where from getting full body inflammation, you get swelling of the brain.
Okay. And on dosing, how does the preclinical dosing compare to the clinical trial dosing?
That's a good question. A lot of laboratory studies will put like 300,000 tumor cells into a mouse, and then they'll give 2 million CAR T cells. And we have certainly done this too. You can see that's not at all what children are going to go through. I mean, we'll give 100 million cells, but not only do they probably have more tumor cells than that, they're spread in multiple areas. And have to move through a much bigger compartment. Because when you give a T cell to a mouse brain, the T cell is the same size as the T cell you'd give to a person. So
Mhm.
you're asking a little T cell to do quite a lot to get to different areas. So it's definitely not a perfect one-to-one, is I would say the take-home message. One of the things that I didn't focus on probably as much as I could have is when we were doing our combinatorial studies, we probably logged like a year of lab work of just dumbing down our CAR T cell doses and finding the dose that wasn't curative, which was like a new exercise. But we had to find a dose that wasn't curative so we could compare other things. And that kind of taught us what's a more reasonable dose where you see a benefit, but don't totally eliminate the tumor. And we've been working with other labs to kind of validate what that number is and working with other teams to try to use that number for future trials so we have a better idea of really what children could expect.
Okay, and specifically on those numbers, how many tumor cells are in a typical DIPG and how many CAR T cells are needed to kill a single tumor cell? Like, is it a one-to-one or?
Yeah, that so I won't quote an exact number of tumor cells because there's a lot of variability. There's children who are very, very sick from what is a small DIPG on MRI. There are children who are walking around looking like you might not know they had a brain tumor with a very large DIPG on MRI, and they have more tumor cells. It's just
Mhm.
maybe not in as dire a situation, or maybe they're not growing as fast at that moment. CAR T cells can engage with a cell, eliminate it, go engage with another cell, eliminate it. But after about 4 or 5 of those interactions, they exhaust, meaning they stop having their ability to kill a cell. They realize, not that they're sentiently thinking this, but they no longer have the ability to keep doing what they're doing and they dissolve basically. So we do think that, there's a certain number of CAR T cells you have to give or a repeated dosing schema, which is why we've done this every 2 week to kind of constantly bathe the area in CAR T cells because we think they last for about 2 weeks.
And those two patients who are still on protocol for Brainchild 03, are they planning to continue indefinitely?
Yeah, it's a good question. I definitely, there's two very, very good adult CAR T cells trials right now that I think both of them are still giving one dose, and that's the end of the trial. And so when sometimes I'm on like a dais with them at conferences and I talk about our kiddos at 90 doses, um, they're kind of shocked by that. But I think that we don't know what the right number is. I mean, I think they're not causing severe toxicity at this point. We have them available. I've always, throughout my career, told children who have otherwise fatal diseases, if you're on a clinical trial and it's going okay, don't jump to another trial. That's just my impression and tends to be what I tell patients because you'll never know if when you leave one, that trial will be available. That trial could close early, that trial could have all types of changes, or you just don't get the same benefit you're hoping for. And it's a little grass is greener is a common feeling. I will say, even as a clinician, you always want your patients to be doing better and look for other options. Um, so I've kind of taken that same approach with these children where I have not encouraged them to do something else, but staying on the trial is definitely a family-driven decision. I've had conversations with both of them about is this the time that you want to come off study? Not necessarily to do something else at this point because they're both doing very, very well. But they've wanted to stay on. They've spaced out their dosing to some degree, but one of them is still really every 2 to 3 weeks, and one of the other ones is every 3 to 4 weeks.
Wow. Okay. And something you said prompted my most burning question because I'm a GBM advocate. But in the adult trials, they saw transient radiological responses, but that really did not translate to a survival benefit at all. Do you think the problem with those trials was the single dose?
I'm not going to use the word problem, which I know you didn't
Okay.
give away in case one of my adult friends is on Facebook watching this. But I think that adult glioblastoma is really, really hard to treat for a variety of reasons. Um, it's just as dire a condition. Um, I think that it's a lot to ask of these therapies to be curative or even beneficial with a single dose. And I think that when you give CAR T cells into the fluid of the brain, there's a circulation of this fluid through the brain, but it's not a closed circuit like the blood. Like if you give CAR T cells into the blood, they go around your body and then they get back to where they started to some degree. Um, when you give CAR T cells in the brain, they might pulse around and move a little bit, but there is a gradient moving down the spine. They do get resorbed at the base. They're not in a supportive environment. If you just put T cells in a bowl of CSF, they're not happy the way T cells are just in a bowl of systemic blood that you would do a study with. So this is just a different situation, and I think that I personally believe, obviously I'm biased, it's how we wrote our trials, to give multiple doses. I do think that they're on the edge, if not already starting to do that. um, I also appreciate that adult glioblastoma patients can also be very fragile in a way that children often are not. And so that the same way we give intensive chemotherapy to 2-year-olds and cure them of malignant diseases, and those chemotherapy doses would absolutely be immediately fatal to an 80-year-old. So it's a somewhat of a different situation, but I think a multiple dosing paradigm, in my estimation, is the best path forward.
Okay. Specifically on B7-H3, what do we know about antigen heterogeneity for the B7-H3 target? Do all tumor cells express B7-H3 to the same extent?
So definitely not all to the same extent. I think I didn't show the slide. Uh, it's either, I think it's Ghidra Conchiti and Dahlia Haidar at St. Jude. Dahlia is now at Children's National Medical Center in DC. I think it's their figure that I think was the most representative, and it showed the degree of B7-H3 staining, and it showed ATRT, ependymoma, medulloblastoma, and DIPG samples that were taken from patients. And compared to other things we tend to target, B7H3, in my opinion, and I think their interpretation, was the highest on the widest range of things. I think Stanford's paper for B7H3 was also called B7H3, a pan-cancer target antigen. So I think that it's more expressed than the other targets. There are some things like GD2, for example, is very highly expressed on DIPG and neuroblastoma, but then relatively low on other things. So I think if this is getting at like what a multi-antigen targeting approach is so I wrote a trial Brainchild 4 where we have a CAR T cell that targets HER2, EGFR, IL-13 receptor alpha 2, which is the target City of Hope is best known for, and B7H3. And that trial is just about to close to filling all the enrollment spots. And it's too early to know too much about it, other than it's mostly been tolerable. And we've been able to make CAR T cells that target four things, which, as far as I know, is the first time that's been done. But even I saw Carl June at a meeting recently, and he was looking at CAR T cells where he did 20 manipulations to the CAR T cells. So the T cells can take a lot more engineering than what we're doing. So I think that targeting more of dealing with the heterogeneity is a solvable problem to some extent. My worry is that it's going to be different for every single tumor. So if you have a medulloblastoma that expresses B7H3, you have a DIPG that expresses B7H3, you give B7H3 CAR T cells, the tumor you have left afterwards is not necessarily going to be the same expression pattern. So we're going to have to learn more about medulloblastoma and its microenvironment. We're going to have to learn more about ependymoma and its microenvironment. If you talk to people like Mike Taylor at Baylor, he'd probably tell you what even is medulloblastoma. They're all different diseases. So it's going to get granular. And I think that what I really, really hope is that we're coordinated in investigating this. Because I hope we don't come out of this in 10 years where everybody's been in a silo doing this. And then we're
Mhm.
like, oh, hey, we all found out the same thing. Like, we just need to be coordinated. But I think the heterogeneity problem is going to be different for different tumors.
Okay, you just covered a lot of the questions because people were asking about the other targets, other antigens. So some B7H3 local regional clinical trials, such as the adult trial at Stanford with Rena Thomas, have asked patients to get to low levels of dexamethasone. Do you feel that dexamethasone negatively affects the efficacy of the intracranial B7H3 CAR T?
So Dr. Thomas is great. We interacted briefly when I was training there. Um, And as far as I know, every CAR T cell trial mandates that you're on a low dose of steroids. What the definition of low is varies. On our trials, I mean, this was probably made this decision in 2017 to do 2 milligrams per meter squared per dose. No, per day divided by two was like the maximum steroids you could be on, and it had to be stable for a week. I don't want to say that's arbitrary, but we just picked a low dose where the main thing is, so children with leukemia, where you are actually getting cancer of your immune cells, steroids are actually a treatment because steroids are so good at killing white blood cells. And also for kids who are on um, steroids for a long period of time, they can actually get, even though their T cell numbers might be normal, they can get infections because their T cells don't work properly. So definitely it is old data that T cells do not like steroids. And so can you make a good CAR T cell product is a question. And then how does it change the tumor is a question. So I think part of it is at least for, are you able to actually generate a CAR T cell product? Meaning, can you take the T cells and make them do CAR T cells and they survive this manipulation? Um, so I think that's probably why they're doing it is to make sure they're getting healthy T cells. The other thing is there's some patients who are on extremely high doses of steroids, either because unfortunately they haven't been able to get off and it is no one's fault, or to be honest, there's some folks who are at maybe less experienced centers or don't have as close follow-up with their primaries and have been on a long-standing high dose of steroids without a weaning plan, and so they just never really came off. Um, In either situation, that becomes very tenuous when you have a tumor that's become dependent on having steroids on board, because eventually steroids stop working. So, and they're one of our rescue systems for when you're giving immunotherapy. So if somebody's been on 4 months of dexamethasone and then they get an immunotherapy and things look worse, dexamethasone is probably not going to help you in the same way. So
Mhm.
there's a couple considerations.
Okay. And I appreciate that you talked about the combinatorial strategies. Have you looked at immune checkpoint inhibitors, anti-PD-1, CTLA-4, LAG-3? Do those have potential for use in combination with CAR T?
Yeah, I would say we have not been leading the charge combining those, but I have some friends who are. I think it is an interesting approach. I think that B7-H3 itself is actually a checkpoint inhibitor. It's not why we target it. We target it because it's there on the tumors, but it's part of the checkpoint family of um, the B7 superfamily of checkpoint molecules. So I think that basically what checkpoints do is they're a way that tumor cells block the immune system from interacting with them to some degree. So modulating that could unlock greater benefit for sure. We know that like one level slightly above that is the tumor immune microenvironment is a problem. It's just a problem that's different for every tumor. So again, is going to be what I think is a tumor specific approach, which I wish wasn't the case because it is going to be slower.
Uh-huh.
Um, and it does mean that maybe we're going to get to a point where different trials have to be for children with different tumors, which is something I'm generally against. Um, but yeah, I think it'll be important to understand checkpoints and the combinational benefit by inhibiting them.
Okay, and then you mentioned the tumor microenvironment. Have you talked about or thought about CAR T cells that attack the other cells in the tumor microenvironment besides the tumor itself, for instance, targeting FAP-A, fibroblast activation protein alpha?
Yeah, that's a great question. I would say that I am not like the world's leading big data scientist on surface expression of things. So there are people better at that than I am who are working on it. It's not something that we've done in the lab, but I think the goal is like you're looking for a therapeutic window. So you could have something that's really, 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 really high on tumor cells or really, really high on the tumor microenvironment. But if it's also relatively high everywhere else, that doesn't help you. Whereas you might find something that is pretty low on tumors, but it's not at all anywhere else, and that's a better option. So some of this gradient is what you're really looking for. But I do think that there is definitely a path forward where the strategy is almost independently how do you target the immune microenvironment? And then how do you attack the tumor? And what is the sequence of those things? And you 1,000,000% can make CAR T cells that have nothing to do with the tumor, but are attacking something else that makes your therapy work better, which is basically what children with leukemia and bone marrow transplant do, right? They get conventional chemotherapy, which is relatively useless against the disease, but is prepping the body in a way for the thing you do next. So, could we make CAR T cells that augment the microenvironment? So then a CAR T cell, and I'm using CAR T cells here, not to say they're the only potential targeted immunotherapy. There's NK cells, there's macrophages, there's other things that you can engineer. There's even things that aren't targeted cellular therapies. But yes, you could independently target the microenvironment before other treatments for sure.
And I remember hearing at SNO, Dr. Manji talk about myeloid-derived suppressor cells. Do you have any thoughts on that? Have there been any lessons learned through this series of trials?
There is a really, really interesting paper by Dalia Haidar, who I mentioned before was at St. Jude and is now at Children's National Medical Center, using, if I hopefully I'm representing this correctly, using different models that they had of tumor cells in mice, some of which had no macrophages or myeloid cells at all, some of which had a lot. Different CAR T cells would have varying benefit against all of them. It was not consistent that if there are no macrophages, every CAR T cell would work better. Some CAR T cells work better if macrophages were present. And to me, that makes a lot of sense. Like, for example, macrophages secrete CXCL10, that marker we're trying to get things to migrate to. So I am more interested in um, making the myeloid cells do what I want them to rather than eliminating them. But I do think that there are ways to make that system better, and to take advantage of the fact that they secrete some things that you want, but probably also eliminate either problematic ones or redirect ones that are working against you.
Okay. Do you have any hypotheses on the underlying mechanism by which the combination of B7-H3 and ONC206 has synergy and induces tumor cell death?
I'm just so excited. Nobody asked me why don't you use ONC201. Okay, for ONC206, I think that there's people like I'm going to focus on the Pete's people. I'm sure there's very good adult people too, but Carl Koshman and Shriram at University of Washington, Javan Nazarian, um, Matt Dunn, a range of people have done really, really good work understanding how ONC206 operates and its metabolomic effects and all those things. So I think what we've seen, and that I hope we're close to publishing, is that ONC206 has these metabolomic changes. Basically, the overall health of the DIPG cell is the way to think of it, but it doesn't seem to affect that pathway the same in T cells. So that's not exactly the answer to your question, but that's why it's an attractive target because again, it's kind of a therapeutic window that affects tumor cells more than T cells. Um, I honestly think they might work well together because they're very independent, working very different ways. So it's like a very non-redundant way to kill the tumor cells.
Mhm.
And that I think is sometimes an attractive approach because you're potentially killing two separate populations of cells. Um, but that probably has a little bit of work left to have a better understanding. I think it might just be that they work very low dose CAR T cells and ONC206 independently work okay, and you put them together and okay plus okay equals very good in a non-overlapping way.
Okay. And another DIPG trial that generated some hope and interest was sonodynamic therapy. Could that be something that could potentially be combined with CAR T in the future?
Yeah, so I think that closed for funding, unfortunately, if I'm remembering right. It's not something that we've worked with directly, but I do know like the UCSF folks, again, Children's National people who have worked on it. Um, I think developing ways to get therapies into the brain better is a very necessary, appropriate approach. And whether that's nanoparticles like Praveen is doing at Rady Children's, or convection enhanced delivery like Mark Sudain is doing, or focused ultrasound like some of these other people I mentioned, I think that they're all valuable. I think with intracranial CAR T cells, we already I mean, I don't want to oversimplify it. And certainly, if my 8-year-old needed to get CAR T cells in his brain every 2 weeks, that would be really hard to go through on top of everything else we're going through. So that's not nothing. But getting the CAR T cells into the brain is a very achievable outpatient thing in our hands. Um, do I wish it was easier or one dose or better? Absolutely. And that's the direction we will move things to once they're working more consistently, more
Mhm.
often. Um, but I do think that we're already getting the T cells into the brain relatively well. So getting CAR T cells into the blood and then using something to get them into the brain, I don't think would be something that we need at this juncture. Now, ultrasounding specifically like the area of tumor could get CAR T cells into a tumor better. Um, it's not a huge priority for what we're doing, but it doesn't mean that it's not important, and I'm glad other people are working on it.
Okay, and getting them to where they need to go. You mentioned the intraventricular and the intratumoral. Could you do both? Have you optimized, kind of whether one or the other or both would work best and
Could you say what two things you were talking about? Sorry, that I missed
like
that.
intraventricular
Oh,
versus
give me.
intratumoral?
Oh, yeah. So, I mean, there's definitely excellent sites that are still doing IV dosing, and Baylor was doing IV dosing before we did anything, and Baylor has a ton of experience, and now is doing a mix of intracranial. Stanford uses a mix. Different people do different things. I think St. Jude is doing only intracranial, but
Mhm.
these trials also sometimes change, so I apologize if I'm misspeaking about what maybe someone's child went through or what the current state is. Um, but it's unknown. They're not mutually exclusive. I mean,
Mhm.
like you could easily imagine giving a CAR T cell directly into a tumor to make a change before you give CAR T cells into another part of the brain or the blood to get there. Um, Getting T cells directly into a tumor just because of tumors or T cells are not as stable as a drug outside of the body. So, like, how do you get them, thaw them? The neurosurgeon places them. There's other inflammation. It's a totally solvable problem, but would take a little bit of extra care in how you're operating that. But yes, you could definitely give CAR T cells more than one way.
Okay. And I'll try to wrap this up with two last questions. Have you seen a change in B7-H3 expression in a patient from the biopsy before treatment versus that? I assume you've done autopsies for some of these children.
Yeah, so sadly, we have had a few autopsies. The trial between running during COVID, the fact that most children who pass away pretty far from their CAR T cell dosing, both temporally and geographically. We have not looked at a lot of pathology on the children that we've taken care of, even though a lot of families are enthusiastic to donate, and we're part of the Gift from a Child network, and certainly want to make that available to every family who's interested in doing that type of generous donation, or child if they're self-directed in doing it. We have not been as comprehensive looking at that as we could be. It's something that hopefully we'll learn more about. We have not put children through a rebiopsy or other things. That's a tough one for me. There are some trials who are doing that. We're not doing that here. Um, I think the issue with all of these things is to the question before of tumor heterogeneity, is if you get a small sample afterwards, how representative is that? Even
Mhm.
when you get an autopsy sample, it's hard to look at all of the disease. But I do think that I'll tell you that we have a paper that's currently being reviewed now where the B7-H3 CAR T cells that migrate better, one of the mice that failed that treatment, the tumor that grew out no longer expressed B7-H3. So
Wow.
that is biologically possible. I
Mhm.
think some of the experience in children and adults has not been that you totally lose the antigen. But again, we're not looking like right after the dose. So the antigen could go away, and by the time we have an opportunity to look, it's back. But it's something we should learn more about.
Okay. And lastly, you're doing the quad targeting. Are we going to see that in adult clinical trials? Do you have any awareness of any plans on
You
that?
know, while we're affiliated with the University of Washington, we're very freestanding pediatric here. So for
Mhm.
adult folks, there's always a limitation on what we're able to do. I think fortunately there's a lot of good adult programs, Penn, Boston, UCSF. Um, so I think that and Stanford, I think that they'll be moving that direction. I think whether you need to target two antigens, three antigens, four antigens, whether it's better to target two antigens or target one antigen and make them migrate better, or is it better to target three antigens and make them migrate better, or target four antigens and then knock out some checkpoint inhibitor? And I think that these are the areas where I hope we're coordinated, because every one of these things is like a 10-year project for each of our labs. Um, and I don't want to be 54 and we're all learning we did the same thing. So I hope that we're going to work together and it will benefit both children and adults.
Okay. All right. I'll let that be the last question and we'll wrap it up. And I just want to thank you so much for the work that you're doing. It's absolutely incredible. I can't say enough good things. I'm such a fan of the work that you're doing, how long you've been at this, how far you've developed this over the years, your inclusive trial designs, the hope that you're giving to children and families. It's amazing.
Well, thank you very much, and thank you for inviting me. It's great to be connected to the community. And if people have more questions, let me know. But thank you for joining and supporting the program and supporting other families and patients.
Thank you.
Yeah. Have a good night, everyone.
Good night.
Thanks all.